Myocarditis is the inflammation of the heart muscle that affects the ability of the heart to pump blood. It could lead to heart attack or stroke. Cases have increased about 59.6% worldwide from 1990 to 2017, and it could affect all ages. The causes include viral infections to adenovirus, COVID-19, hepatitis C, Epstein-Barr, bacterial fungal infection. Also, repeated attacks of rheumatic fever, fibrosis, trauma, ischemia, or cardiac toxicity. With symptoms, we can see shortness of breath, irregular heartbeat, chest pain, and flu-like symptoms. For diagnosis, they could utilize electrocardiogram, chest x-rays, and biopsy of the myocardium to evaluate for the presence of B cells and T cells, as well as macrophages. For treatment, they could use antibiotics specific to the viral infection, non-steroidal anti-inflammatory drugs to suppress the innate immunity, and corticosteroids to suppress the adaptive immunity to decrease myocarditis. Myocarditis is the inflammation of the heart muscle. In myocarditis, the first system to respond is the innate immune system, which is stimulated by the presence of virus or trauma. Among the responders, we have macrophages, mast cells, and T helper type 1 cells, which can secrete different cytokines. We can see pro-inflammatory cytokines like interleukin 1 beta, interleukin 4, 12, 23, 5, 6, and 8, as well as TNF alpha. And we can also see anti-inflammatory cytokines like interferon, gamma, interleukin 10, interleukin 23. Then we have the adaptive immune system, which is stimulated by the innate system. And during this process, we see pro-inflammatory cytokines like interleukin 4, 12, and interleukin 1 beta, again, and IFNG gamma. In our study, we found that during the innate immunity, interferon gamma can actually protect against myocarditis by suppressing mast cells degranulation, which usually secretes TGF beta, interleukin 4, and interleukin 1 beta, which are pro-inflammatory, therefore reducing fibrosis that can potentially lead to myocarditis. We also found that during the adaptive immunity, suppressing Th17 cells can reduce myocarditis. This is done so by blocking the high mobility group BOX1, which is a protein secreted by macrophages during the adaptive immunity. So the way that the high mobility group BOX1 works is by binding to two like receptors like 4 and 2, as well as RAGE, which can trigger cytokines release like interleukin 4, interleukin 12, interleukin 1 beta that are pro-inflammatory, as well as TGF beta and IFN gamma. The purpose of this graph was to observe if myocarditis and fibrosis would be increased in mice with interferon gamma deficiency. As we can see in figure 1A, there is an increase of myocarditis in mice that had interferon gamma deficiency. We see that there was no increase in the control or the mice with interleukin 12 receptor deficiency. And we can also see that in figure B, with fibrosis, mice that had interferon gamma deficiency also presented an increase in fibrosis, and there was no increase in mice with interleukin 12 receptor deficiency or the control for fibrosis. So this concludes that interferon gamma does actually reduce myocarditis by reducing fibrosis, by reducing the viral replication.
So the goal of this graph was to test for the levels of interleukin 1 beta in the hearts of mice, which is a pro-inflammatory cytokine. In this figure, there are multiple types of mice used. This study involved mice that were considered a knockout group and a control group. A knockout mouse is a laboratory mouse in which researchers inactivated an existing gene. The knockout groups included interferon gamma and interleukin 12. We also have two control groups, one uninfected and one infected group. The mice were infected with Coxsackie B3 virus, CB3 for short. So CB3 closely resembles human myocarditis in the symptoms. The uninfected mice were not induced with CB3, and the mice that were infected with CB3 were induced on day 0. Hearts from all mice were collected and analyzed on day 35. The levels of interleukin 1 beta were tested using ELISA kits, and it was found that interleukin 1 beta levels increased in interferon gamma deficient mice with induced myocarditis. By looking at the results, it was found that interferon gamma protects against increasing levels of interleukin 1 beta and CB3 induced myocarditis. The goal of this study was to measure the levels of interleukin 4 in the hearts of mice. Similar to the previous graph, there was a knockout group and a control group. The knockout mice were deficient in interleukin 12 and interferon gamma. There was also a group that was induced with Coxsackie B3 virus, which causes similar symptoms to myocarditis. This was considered the infected group. The uninfected group was not induced with CB3. The infected mice were induced with CB3 on day 0. All hearts from each group were collected and analyzed on day 35. Levels of interleukin 4 were tested using ELISA kits. By looking at the results, it was found that levels of interleukin 4 increased significantly in interferon gamma deficient mice with induced myocarditis. As you can tell, looking at the graph, both groups of interferon gamma deficient mice had large levels of interleukin 4, but the mice that were infected with CB3 had a little bit larger level of interleukin 4. Therefore, it was found that interferon gamma protects against increased levels of interleukin 4 and induced myocarditis.
Okay, this is the start of our adaptive graphs that are testing the cytokines IL-6 and TGF-beta levels in the serum of infected mice. These mice were infected with experimental autoimmune myocarditis, EAM. The goal of this was to test the levels of IL-6 and TGF-beta and the effects HMGB1 blockade would have. The way that HMGB1 blockade is working is by having the antibodies attached to HMGB1, which therefore blocks the pathway that usually causes the secretion of cytokines, such as IL-6. In the figures, we have C, which is our control, E, which is our induced myocarditis, and E-B is our induced myocarditis mice with the HMGB1 blockade. The samples were taken after 21 days and show the results of the TGF-beta serum and IL-6 are significantly decreased after the HMGB1 blockade was induced. Okay, this is the start. This next figure is the IL-17 and interferon gamma in the serum of mice that were infected with the myocarditis. The goal in this figure was to test HMGB1 blockade, and it evaluated the serum and concentration in the blood of the 3 different samples, once again being C control, E induced, and E-B induced and blocked. The results of this were that IL-17 levels were decreased after HMGB1 blockade was introduced to the infected mice. And as you can see with interferon gamma, there was no change. This
slide is covering our discussion and conclusion of our innate portion of our presentation. The largest finding in this experiment was the role of interferon gamma. Through the experiments involving mice, it was found that interferon gamma plays a role in myocarditis, fibrosis, and cytokines. Being deficient interferon gamma increases myocarditis and fibrosis. It also increases levels of interleukin 1 beta and IL-4. The figure on the left is including images of fibrosis and inflammation in the hearts of the mice. This was taken from the article. Looking at the figure to the left, section A is a control group that was uninfected with CB3. Section B is the infected control group. And group C is mice deficient interleukin 12, and group D is mice deficient in interferon gamma. The blue portion in the images represents scarring and damage to the heart. As you can tell, group D, which is deficient in interferon gamma, has the greatest amount of damage. Through this article, it was found that interferon gamma protects against the severity of myocarditis and fibrosis. It also reduces the amount of interleukin 1 beta and interleukin 4 cytokines. It also affects the development of Coxsackie-B3 virus.
HMGB1 is released from macrophages in the tissue and will signal on other immune cells through a receptor such as toll-like receptor 4. Secretion of IL-6, which drives Th17 cells, is the result of the HMGB1 pathway. And what the blockade is doing is attaching a monoclonal antibody, which is ultimately blocking HMGB1 from being able to attach to the receptor, otherwise known as neutralization. This ultimately reduces IL-6 and IL-17, which is blocking the Th17 cell expansion, therefore decreasing myocarditis. Overall, what this has told us is that the HMGB1 blockade reduces the number of inflammatory cytokines such as IL-6 and IL-17, as well as decrease myocarditis using the antibody specific for HMGB1 to block its function. Blocking its function suppresses the Th17 cell expansion in the heart. If the blockade is not there, we learn that HMGB1 will be released from the macrophages in the tissue and signal for other immune cells to secrete IL-6, driving the CD4 T cell Th17.